The genotype is a term used in genetics that is used to refer to the genetic composition of a cells or of a microbe consisting of heritable genes. So the physical characteristics of the cells called phenotypes are actually the results of the expression of these genes. The phenotype is refer to all the observable traits in organisms, so the actual and the expressed properties of the genotype is called phenotype. Genetic information flows from DNA into protein, the substances that gives an organism its form. So this flow of information occurs through a sequential process of transcription, which is from DNA to RNA, and then translation from RNA to protein. So this genetic flow is called the central dogma. So the pathway from from DNA to RNA and then to protein is called gene expression. So this central dogma process is actually conserved in all cellular forms of life. So the flow of the genetic information is passed from one generation to the next one. So DNA stores the genetic information, and this information can be duplicated by replication process, and then it is then passed through from one generation to the next generation. The gene expression. So in DNA is divided into genes. So in this process of gene expression, the transcription process will yield a ribonucleic acid RNA. So from DNA to RNA is called transcription. So this process will actually copy specific genes. And then the next process is called translation. So translation will use the information in the RNA, the mRNA, messenger RNA, to synthesize a polypeptide. Polypeptide means a chain of amino acid peptide or into protein. So this activity not just involves mRNA, but also involves the tRNA, transfer RNA, and as well as the ribosomal RNA. So this is a figure that shows the different processes of or the different phases of the central dogma. So how DNA makes RNA and then makes protein. So the information will flow from DNA to RNA and then to protein. So what we have in the first stage is we have the DNA where all the information is stored, right? So DNA is nucleotide in the form of nucleotides. So we have the 4 nucleotides, all information is stored in those 4 letters, A, T, C, G, and this A, T, C, G will then perform transcription, will go through transcription, and then this DNA is transcribed into mRNA. So where mRNA is the carrier of all the information. So when we actually, in the transcription process, so A will pair with U, so we have uracil, T will pair with A, G will pair with C, all right? So we don't have T in mRNA sequence, in RNA sequence. After that, the RNA will go through translation. So those 3 letters of the RNA will form together, will form a codon. So that 3 letters codon will actually translated into amino acid. All right. So the codon, for example, UUA will be translated into leucine, CAC into histidine, GGC into glycine. So these amino acid will then link together to form a polypeptide or protein. So this protein is actually the active machinery, active cell machinery. So DNA in the cells will go through these processes, which are replication, transcription, and translation. So replication does not involve in the central dogma of genetic flow. So replication is a process of duplicating or replicating the DNA so that the genetic information is passed through from one cells to another cells. All right. So on the other hand, the process of genetic flow involves transcription and translation. So transcription is the process of DNA being converted into mRNA that carries the information, and then the mRNA is then translated into protein. So these protein or gene expression is very important for the cells in order for its structure and the function of the cells. So without the success of this gene expression, um, so the phenotype will not be shown, or everything is failed in the system. First, let's take a look at the process of DNA replication. So DNA replications require these ingredients. So you need a DNA template, you need primers, a DNA polymerase 3, you need ligase, DNA primase, helicases, and a single strand DNA binding proteins. So what are the functions or what are the functions of these ingredients? So we're going to have a look at this on the next slide. So a DNA template is actually a preformed pattern copied from the old DNA, and then the old DNA and the new DNA is not covalently connected. Okay, and then next we have primers. Primers is a short sequence or a strand of nucleic acid sequence that serve as the starting point for DNA synthesis. Alright, DNA polymerases only add new nucleotides to an existing strand of DNA. So without this existing strand of DNA, without the primer, so DNA polymerase will not add it new will not in new nucleotides. So the replication will start at the 3' end of the primer and copies the opposite strand. DNA polymerase is an enzyme that synthesizes new DNA from 5' to the 3' end. So the enzyme add a nucleotide onto a pre-existing 3' hydroxyl group. So you're going to look at the process clearly after this. The enzyme that responsible for the process in the DNA replication is the DNA polymerase 3, so where it actually sticks to a primer on an open DNA strand, and then it will build the second strand from 5' to 3' end. For replication, so you also need DNA helicase, so the one that actually unwind the helix structure of the DNA. So this is the enzyme, so DNA helicase. So the other ingredient is called single strand DNA binding proteins. So these proteins bind individual strands of DNA, double-stranded helix. It will aid or help helicase to open up the DNA double-stranded helix into single strands of DNA, and then it also helps stabilizing the unwound single strand conformation, preventing tangles for the DNA strands. We also have primase. So primase is the enzyme for the RNA synthesis. So it provides a starting point of RNA or DNA for DNA polymerase to begin synthesis of the new DNA strand. So lay down initial RNA primers so that DNA polymerase can work. Okay, the next enzyme is called DNA ligase. So this DNA ligase will actually glue together pieces of DNA created by DNA polymerase 1 and 3. So also it then built the second strand from 5' end to 3' end. This is basically how the process of replication, DNA replication looks like. We have the double-stranded DNA that is open, right? So we have the first strand here is the 5' end to the 3' end here, and then we also have the 3' end to the 5' end, right? So always the other strand, the opposite strand is always in the opposite directions. And just for you to remember that the DNA polymerase will synthesize from 5' to 3'. So it will actually use the 3' end to the 5' end strand as a template. Okay, so what we have here is the helicase enzyme that actually unwind the double-stranded DNA, right? And then so we have the opening of the double-stranded DNA, and then that we call it as replication fork. So replication fork is where the replication start. Okay. And then we have the DNA primase. DNA primase is the starting point of the RNA or DNA polymerase to begin synthesis of the new DNA strand. And then we have the primer, a short sequence that will actually start the replication. And then we have DNA ligase that actually will glue together the pieces of DNA created by the DNA polymerase. Okay, and we have the DNA polymerase, and then we have this term called the leading strand, and then we have the term called lagging strand. So, and also not to forget, we have the single strand binding protein that helps in this to stabilize the unwind single strand DNA so that it won't be tangled. Okay. So first, I would like to explain on how the replication process happen at the leading strand. All right, so the DNA strands, as I mentioned, have an anti-parallel nature. So where one strand will run from 3' to 5' end, and the other one will run opposite from the direction. So DNA polymerase can only synthesize new strands of DNA in the direction of 5' to the 3' end. So in order DNA polymerase to do this, it must read the template from the 3' to the 5' end. So this is actually the easiest way for a process of replication, where it is quite direct how the DNA polymerase synthesize the new strand. All right, so I'll explain to you how the process. Number one, it involves helicase. First, the DNA double helix is opened by the helicase into two individual strands, and then topoisomerase will actually relieve the tension further down in the double helix. And then what we have then, the stringer strand binding proteins will help to stabilize the unwound parental strain of DNA in order to prevent them from reconnecting together. And then we have the RNA polymerase is added to the leading strand at the complementary base of the primase. So the leading strand is continuously synthesized continuously in the direction of 5' end to the 3' end direction by DNA polymerase. So DNA polymerase, after we have the primase, DNA polymerase will attach to the primer actually, and then sliding clamp stabilizes the DNA polymerase. So DNA polymerase will actually move down the leading strand toward the replication fork. So you can see here 5' to 3', and then on the leading strand we have 5'. So the DNA synthesis start from 5' to the 3' end, right? So it is actually moving towards the replication fork. All right, so that is how the process of data replication on the leading on the 5' to the 3' of direction of the DNA. So using the leading strand process. As I mentioned earlier, DNA polymerase can only create new DNA strands from 5' to the 3' end. Okay, so therefore, when 5' to the 3' template strand is being replicated, where the new strand must run opposite in the 3' to the 5' end direction, so the new strand actually cannot be synthesized in a continuous fashion as the leading strand was. So to overcome this challenge, additional steps are needed to replicate the 5' to the 3' template strand. So where this newly synthesized strand is known as the lagging strand. In order to synthesize the lagging strand, DNA needs to be broken into smaller segments known as the Okazaki fragment. Because of these multiple segments, the lagging strand is also known as discontinuous strand. So it is not continuous strand as compared to the leading strand. So by creating these multiple segments, DNA polymerase is able to synthesize a small portion of a new DNA strand away from the replication fork towards the Okazaki fragment direction. So the process involved are the same. So the DNA double helix is opened by helicase, and then we have the SSBP, single-stranded binding protein, will stabilize the RNA DNA strand. Primase will add in the RNA primer to the lagging strand, and then DNA polymerase bind to the primer and creates a short fragment of newly synthesized DNA from 5' to 3', synthesizing the opposite direction of the replication fork. So this is the first Okazaki fragment. So as helicase further unwind the double helix, the replication fork moves down the strand. So another primer is added to the fork. DNA polymerase then attaches to this primer to synthesize a second Okazaki fragment in the direction of 5' to the 3' direction away from the replication fork. So once the DNA polymerase reaches the first Okazaki fragment, DNA polymerase 1, so now we have a new DNA polymerase, right? So removes the primer and replace it with the proper complementary bases. So, and then DNA ligase will connect the segment of DNA by closing the sugar phosphate backbone, and then the two segments are now connected into single strand. So this process repeats as the replication fork continues down the length of the DNA. Okay, so that is how actually the replication on the opposite DNA template on the direction of 3' to the 5' end, 5' to the 3' end, sorry. So scientists has successfully captured the process of chromosome replication in E. coli here. So what you can see here is number one, the bacterial chromosome, the parent copy of it. All right, and then start with the origin of replication. the replication fork, whereby one single strand of DNA, the blue one, will create a new DNA fragment, so that will be replicated. So the purple one, so that is a new strand of the DNA. All right, then now you have from two copies, one DNA into two strands of DNA strands, right? So now we have from one copy of chromosome into two copies of chromosome, right? So, and then we have at the end of the process, there'll be a termination process where the replication will end, and then finally you have two copies of the chromosome. So please note that you have different colors represent different strands. So number one, the parent strand and the daughter strand together. So in the process of replication, so the replication fork will actually moves along the parental DNA. So the original strand and the newly synthesized daughter strand will rewind. This newly produced double-stranded DNA contains one original, which is called the conserved strand, and one newly synthesized strand, right? So that means so that's why we the term of replication is called semi-conservative replication. Semi-conservative replication describes the mechanism of DNA replication in all known cells. So this process is known as semi-conservative replication because of the two copies of the original DNA molecule are produced. So each copy contain one original strand and the black one, right? So that is the original strand, and then the other one, and also one newly synthesized strand. So the red one is the newly synthesized strand. So the structure of DNA suggested that this is found by Watson and Crick, that the structure of the DNA suggested that each strand of the double helix would serve as a template for the synthesis of a new strand. To end this topic on the replication, I just would like to present some fun facts about replication. Number one, DNA replication is an amazingly accurate process. All right, so only one wrong base added in every 10 to the power of 10 bases incorporated. All right, mutation do happen, but it's in very small percentage. The DNA replication process will actually involve a proofreading process as well. So DNA polymerase is able to proofread the sequence of the nucleotides in the DNA. Also, it evaluates if proper base pairing structures are formed. If not, then it will be excised and replaced to the correctly base pairing structure. All right.